Now proteins, as we all know, they are the polymer of amino acids. And what do we mean by the term the polymer of amino acid? It is just a linking of one type of amino acid with another type of amino acid and so on, right? So this is mainly the term called as the polymer of amino acid. And generally, the proteins are mainly heteropolymeric in nature. It means that none of the amino acid in the sequence of the protein is of similar in nature. Okay, it is not made up of a repeating unit of a single type of amino acid, right? It is made up of a different type of amino acid. That is what we mean by the term heteropolymer, right? All of the proteins are interestingly heteropolymeric in nature. But in the case of cellulose, what you people have studied, you can see that in the case of cellulose, it is mainly a it is mainly a homopolymer, right? It is mainly a homopolymer. And which sugar unit was mainly observed in the case of cellulose? It is mainly the glucose, right? It is a repeating unit of glucose molecule that mainly results in the formation of the cellulose, right? The same molecule is also involved in the formation of, as we all know, that is the glycogen and also the starch. So hence we say that the proteins are mainly heteropolymeric in nature and they are polymer of amino acids. Now we have also discussed in the previous classes that when we look into the amino acids, right, when we look into the amino acids, that is mainly about the properties of amino acids, we had mentioned that there are around 20 different type of amino acids. Okay, there are 20 different type of amino acids. Some of them are neutral, some of them are charged, that is acidic, or what we can say basic, that is positively or negatively charged. And similarly, we had even mentioned that some of the amino acids are even aromatic in nature, right? Yes. So this is also one way of classification based upon the type of functional group these amino acids mainly contain, that is the R group. Based upon the R group we had mainly classified, right? But we can also classify the amino acids based upon the need, that is necessity, or the ability to synthesize these amino acids in the body or not, right? So based on this criteria, that is ability to synthesize the amino acids, right? So here we are having first class, it is called as the essential, okay, it is called as the essential amino acid. And one more we are having is the non-essential amino acids, right? Non-essential amino acids. Now, as I mentioned that this classification is mainly based upon whether the body can synthesize or not, right? So essential amino acids are the amino acids where they are not produced or not synthesized in the body. They are not synthesized in the body, right? Hence they are called as essential amino acids. And the only way to obtain them into our body is mainly through the nutrition. Okay, mainly through the nutrition, that is mainly through diet. Whatever food we eat, that is the only way through which we can actually obtain these essential amino acids. So on the contrary, we are having non-essential amino acids, as we had mentioned over here. Now it is exactly reciprocal or opposite to that of the essential amino acids, that is they can be produced by the body. Okay, they can be synthesized by the body, and there is no compulsion, there is no necessity that these amino acids should be determined through the, or should be obtained through the nutrition or through the diet. Okay, that is what we mean by the term non-essential amino acids, right? So ultimately these different 20 types of amino acids are mainly involved in the formation of a polypeptide, or that is the proteins. Okay, and amino acids, they may be either essential type of amino acid, or they may be non-essential type of amino acids. Now moving on, we have to consider two important proteins. Of course, we do have a table where we will be discussing about different type of proteins, examples for the different type of proteins, right? So the first one we are having is the collagen, and one more we are having is the rubisco. Okay, now both of them are considered to be the most abundant proteins. But interestingly, collagen is one of the most abundant protein only in the animal world, right? Only in the animal world or in the animal kingdom, collagen is considered to be one of the largest or most abundantly found protein. But in the case of rubisco, it is a protein that is mainly found in the plants. Okay, it is a protein. More specifically speaking, we can say that rubisco is actually an enzyme. Okay, it is mainly an enzyme that is mainly found in the case of plants, and we consider the rubisco as the most abundant protein in the terms of biosphere. Okay, in the terms of biosphere. So ultimately, the major organisms that you find on the planet Earth are none other than the proteins. Plants. Okay, so plants are ultimately the highest number of individuals that you see on the planet. Hence, we consider that the rubisco is the most abundant protein in whole of the biosphere. Okay, of all the organisms, of all the different type of living organisms, we see that rubisco, that is an enzyme, is the highest abundant protein you find in the case of biosphere, right? And full form of rubisco, as we all know, it is actually the ribulose bisphosphate carboxylase oxygenase, right? Yes. So you will study about the rubisco enzyme more clearly in your photosynthesis chapter, that is the C3 reactions and C4 reactions. You will mainly study about the rubisco enzyme. We will encounter why this is a carboxylase and also oxygenase. It is having affinity towards both the carbon dioxide and also the oxygen. Hence it is named as carboxylase and oxygenase. Right? So here we are having a table. Let me erase this. Yeah. So here we are actually having a table that briefly discusses about mainly the type of protein and what is their respective functions. Right? So now if you look into the first abundant protein in the case of the animal kingdom, that is the collagen, you can see that it is actually acting as an intercellular ground substance. Again, I have to point out that this whole table is of importance. Okay, lot of questions have appeared in your CET examination, that is Karnataka CET examination, and also in your NEET examination regarding the type of protein and the respective function of that particular protein. Okay, hence you don't have to ignore, okay, don't even considering or thinking of ignoring this particular table over here. Okay, so collagen is actually acting as an intercellular ground substance. It is acting as an intercellular ground substance. Okay, so you will hear about collagen a lot in the case of a tissue, in the animal tissue called as the areolar tissue. Okay, so you will mainly hear about it in the case of areolar tissue. Right? So in the structural organization of animals, we will study about it. Now, moving on, we are having an enzyme, okay, that is called as the trypsin. Okay, now trypsin is actually a protease. Okay, it is mainly what? A protease. Now, what do you mean by protease? Protease is something that is involved in a degradation of proteins, right? It is mainly involved in a degradation or digestion of proteins. Okay, if the proteins are needed to be digested, if the proteins are needed to be degraded, broken into smaller pieces, then proteases are the enzymes that are involved in this mechanism. Okay, and one such protease that you're going to look over here is the trypsin, which is exactly as I said, it is an enzyme. Okay, yes. The next one we are having is the insulin, right? And insulin is actually a hormone, and interestingly, it is a protein hormone. It is a protein hormone. Okay, and we all know the function of the insulin. The function of insulin is mainly to reduce the blood sugar levels. Okay, mainly the blood sugar levels. Yes, the blood sugar levels are mainly brought down, okay, by the action of the insulin. And the next we are having is the antibody, right? Now antibody, it is also called as the immunoglobulin. Okay, and the antibodies are also abbreviated as AB. Okay, the antibodies are also called as the immunoglobulins, right? So yeah, we are having IgA, okay, IgG, IgD, IgE, right? And also last one we are having is the IgM. All of these antibodies are actually the proteinaceous in nature. All of these immunoglobulins are mainly proteinaceous in nature, and these immunoglobulins or the antibodies are mainly involved in defense mechanism. Okay, they are mainly involved in the defense mechanism, that is they mainly fight against the infectious agent. That is the function that they have mentioned over here. Okay, here they are mentioned over here. And the next one we are having is the receptor, right? Receptor, and it is mainly involved in a sensory perception, right? So examples mainly include the smell, taste, okay, and also we are having hormones. Yes, hormones ultimately bind to the receptor. For example, we are having insulin over here, right? So insulin is actually a protein hormone, and the receptor for this insulin is present outside the cell itself. They are called as extracellular receptors, and these receptors are also made up of proteins only, right? So binding of the hormones to the receptors will actually ignite or initiate a secondary response inside the cells. Yeah, that will study in the human physiology. Anyway, so they are involved in hormone binding and response mechanism. Sensation such as the smell and taste are mainly because of receptors, and even receptors are proteinaceous in nature. And finally, we are having the GLUT. Try to remember this. It is one of the most important of all in the tables, okay? Because past, that is in the previous examinations about the GLUT4, they have mentioned few questions. I have seen few questions that have mentioned about the GLUT4. Okay, so now the GLUT4 is actually a carrier protein. Okay, now GLUT4. The term GLUT4 mainly means it is a glucose transporter. It is mainly a glucose transporter. Okay, and as the name indicates, glucose transporter, it is mainly involved in uptaking or allowing the glucose molecules, okay, glucose molecules into the cells, okay, into the cells. That is what these GLUT4 are doing. And remember, the GLUT4 is actually, it is, as I mentioned, it is a carrier protein, right? It's actually a carrier protein, and as we all know, carrier proteins are mainly present in the cell membrane. Exactly, remember this. They are mainly present in the cell membrane. Yeah. So, and also if I have to recall some important points about carrier proteins, they are mainly involved in the facilitated diffusion, okay? And any carrier proteins do not require ATP, okay? No ATP is required for transporting of molecules from one region to another region, and generally the carrier protein will allow the movement of molecules down the concentration gradient, right? That is from region of higher concentration to the region of lower concentration. Right. So here in this table, they have mentioned mainly about a few important proteins and their respective functions. Now, we will look into the structure of proteins. Let me erase this. Yeah, so let us look into the structure of proteins. Now, when we are looking into the structure of proteins, you see that there are four levels of protein organization. Okay, protein structure or protein organization. Yes, 4 levels. You see that proteins are actually organized. Okay, so the 4 structures that we have to consider over here, the first one we are having is the primary structure, the next one we are having is the secondary structure, the third one we are having is the tertiary structure, and finally we are having is the quaternary structure. Okay, the quaternary structure is the highest of all. Okay, it is the highest level of organization. Okay, the simplest level of organization, it is mainly the primary structure. Okay, so let us look into what these structure actually mean. The first one we have mentioned over here is that they are mainly the primary structure. The first structure we are having is the primary structure. That is, they are mainly sequence of amino acids. Now, what do you mean by sequence of amino acid? See, in the beginning of the class, we mentioned that if I consider one amino acid, okay, I will link it with another amino acid, and similarly, I will link it with another amino acid, and so on, right? So now the sequence of amino acid, which amino acid is present at the first position, and next to it the second amino acid, next to it we are having is the third amino acid, the next amino acid we are having is at the fourth position. Okay, so if we look into the sequence of amino acid one after the other, then we call it as the primary structure. Then it is called as what? Primary structure. Okay, and interestingly, if you consider any protein, even the proteins also exhibit polarity. Okay, even the proteins also exhibit polarity. Okay, it means that one end is different from the another end, right? So generally you see that we see that the beginning end is called as the N terminus. Okay, it is called as the N terminus. And the end part, this is the first part, the end part is called as the C terminus. It is called as the C terminus. Now, why do we need to refer the first part, the beginning part of the of the protein as the N terminus and the end part of the protein as the C terminus? Because we can recall from the structure of the amino acids, right? Always we are having the amino group, the alpha carbon, the carboxyl group, and here we are having hydrogen. Okay, and here we are having is what? The R group, right? So what I will do is that I will link another amino acid to this, right? So similarly we can write it NH2 SCCO, and we are having a N terminus, NH. I will write the H over here. Again C and CO, again NH. Okay, let me write the H and R group, similarly H and the R group, and similarly C, CO, and so on, right? Yes. So you can see that the first part we are having is the amino terminus. Okay, amino terminus, and the last end we are having is the carboxy terminus, right? Hence we refer the amino terminus as N terminus, okay, and the carboxy end is referred as the C terminus. Okay, so this is a general structure of the protein, or a linear protein in a nature or structure. So that is why we see that we say that we are having polarity in the case of the in the proteins also. Now moving on, the second level of organization, that is after the primary structure, we have even mentioned about the secondary structure, right? So alpha helix and the beta plated sheets are actually considered to be the second level of organization, that is the secondary structure. Okay, so now what do you mean by alpha helix? It is a coiled spring-like nature. Okay, this is actually called as the alpha helix, right? Yes, and it all depends upon the direction of coiling. Okay, either the coiling may be right-handed side, or either the coiling may be left-handed side. Okay, generally in the nature, all of the proteins that are known to occur are occurring in the right-handed alpha helical structure only. Okay, and the next one we are having is a slightly variant of the alpha helix, that is called as the beta plated sheets, where amino acids are arranged in the form of a sheet like nature. Okay, in a parallel sheet like nature. Okay, so the main difference exists mainly in the type of bond formation. Okay, the hydrogen bond formation that is resulting in the alpha helix or the beta plated sheets. Okay, yes. So the next third level of organization we are having is the tertiary structure. Okay, so in the case of tertiary structure, what happens is that the protein actually folds into a loop-like structure. Okay, this is the N terminus and this is the C terminus. Okay, so what happens is that the protein actually folds into a loop-like structure. Okay, so folding property of the polypeptide is considered to be the formation of tertiary structure. Okay, so along with the folding of polypeptide, you also see that there is one more important factor that is resulting in the formation of tertiary structure, that is called as the disulfide bridges. Okay, disulfide bridges or disulfide bonds. They're also called as disulfide bonds. Okay, and interestingly, for the formation of the disulfide, the name itself indicates two sulfur molecules. Okay, hence it is called as disulfide bridges. Okay, and disulfide bridges are important for the activity of the protein or polypeptide. So you can see that the amino acid which is mainly involved in the formation of disulfide bridge or disulfide bond is the cysteine. Okay, now cysteine is an amino acid which is mainly containing the sulfur. Okay, it is mainly containing the sulfur. Okay, hence we can say that it is cysteine that is involved in the formation of the disulfide bridges. Okay, so disulfide bond formation and folding of protein is folding of the polypeptide is important for the formation of the tertiary structure. Okay, so ultimately the biological functioning of the protein is determined by the tertiary structure. Remember, okay, the ultimate functioning, that is whether the protein is actively functioning or it is not functioning or it is remained in the inactive state, that is determined by which structure, which level of organization, that is the tertiary structure. Right? And finally, the next level of organization that we are having is the quaternary structures. Right? Now, what happens in the case of quaternary structure is that one type of polynucleotide, it binds or assembles with another different type of polynucleotide, and both of these will become into what? A functional unit. It will become into what? A functional unit. It becomes into a complete functioning protein. Okay, an excellent example for the quaternary structure that we know is the hemoglobin. Okay, it is the hemoglobin that we are all very much aware. Okay, we say that hemoglobins are mainly made up of two polypeptide chains, that is alpha and beta. Okay, alpha is present in 2 copies and beta is present in 2 copies. Hence we say that alpha 2 beta 2 is actually the formula for the hemoglobin. Okay, and as we all know, hemoglobin is mainly involved in the transportation of oxygen, okay, from blood to the tissues, and also carrying the carbon dioxide from the tissues to the lungs. This is the main function of hemoglobin, right? So these are the levels of organization or structure of the proteins, right? I hope we have made the points quite clear over here. Okay, so again, the structure of proteins is an important part that you people have to remember. Okay, so this is the part of this part of the discussion where you can expect the questions kind of to be true or false or incorrect kind of statements. Which of the following is a correct statement or which of the following is an incorrect statement? This is what kind of questions you may expect in your examination. Okay, so for this you need to understand the concept of proteins quite vividly.